Okay. So you can see that okay?
Yes, it is coming. Yes, yes, we can see your presentation, right?
Okay. Is the connection okay? Are you able to, is everything happening in real time here, or is there delays of any sort?
No, it is on real time. You can just switch it to the presentation mode and you can start. But now we have 4-5 minutes, so we can wait for some more people to join. Maybe. I'm really
Okay.
thankful. I'm really thankful that you agreed and you came to presentation.
Yeah, I was happy to do it. I'm glad to be invited. Still think I think I'm going to move to a different spot because I think the connection in this part of my house is not the best place to be. I'm afraid that you may lose me. So I'm just going to move to a different room.
Sure, sure. So Dr. Richie is also here.
Oh, is he? Oh, great.
Richie, are we audible to you?
Yes.
Hi.
And my volume is appropriate. You can hear me okay?
Yes, absolutely. So Dr. Chauhan, are you online? So Dr. Chauhan?
Hi, Pratap. Hello, Dr. Daniel Kenny. How are you?
I'm well.
So this is
Thank you.
Dr. Chauhan from Department of Microbiology. I'm colleague of Pratap. So
now the time is actually 16:30. So I'll request Dr. Chauhan to introduce our guest.
okay, so shall we start now?
Yes, we can start. Please take up.
Okay, so good evening, guys. Welcome back after the tea session. So this will be our last session of the day of the webinar. And the speaker of this session is Dr. Daniel J. Kenny, who is currently chair, Department of Pharmaceutical Sciences, and also is an associate professor of medicinal chemistry at School of Pharmacy, Temple University, Philadelphia. So he received his BS and PhD from Temple University and did a postdoc from School of Medicine, Washington University. And after that, he has also supervised 5 PhD candidates, published more than 35 research articles, 65 conference presentations, and 5 patents to his credit in the field. And he has handled more than 15 research grants, visited several international universities on various assignments, and members to several committees of his institute. And his current research interests includes synthesis, characterization, and evaluation of novel ligands for muscarinic acid, nicotinic, serotonin, and retinoic acid receptor subtypes. So today he'll be talking about, as you can see in his presentation, about GPCR ligands. So I welcome Dr. Daniel Kenny to this webinar. Thank you so much for your presence, and we are excited to listen to you for next 1 hour. Thank you so much. It's over to you, Daniel.
All right, thank you very much. Thank you for that introduction. I'm trying to share my screen here, and it's not, doesn't want to cooperate with me. I thought I had it.
Just make it into the presentation mode and you can start your presentation.
It says you're presenting to everyone, but it's not showing me anything.
You just go to your presentation. Go to your PowerPoint. Go to your PowerPoint. Just open your PowerPoint. Let's maximize it.
Oh, you can.
We can see it, what you are doing. Yes, it is
Okay,
now.
so now you can see that?
Yes, we can see that.
All right, well, good. All right, good. Thank you for the invitation. It's really a pleasure to be here to talk about some of the research that's been going on in my laboratory at Temple University School of Pharmacy. I'm going to be talking about a couple of different projects, but I'm going to speak about them all in connection to one particular set of ligands. So the title of the talk is Lead Modification Approaches in the Design of GPCR Ligands in an Academic Drug Discovery Environment. Or, as I say here, academic drug discovery, what a long, strange trip it's been. And that's one of the things I want to talk about is the fact that we have a new drug discovery center. It's no longer new, but in my mind, it is new. I've been at Temple for about 25, 26 years. The center's been there for about 10. And that's one of the things I want to talk about is how that the introduction of the Mulder Center for Drug Discovery Research has changed the way I do things in my lab. So this is a picture of some of the people that have been in my lab over the years, primarily PhD candidates. Dr. Bandari is shown here, was a student in my lab some years back, and some other folks that I won't get into. But these are just some of the projects that were mentioned just a moment ago. What I'm going to talk about today is the first one, ligands for muscarinic receptors, and then I'm going to move from muscarinic receptors into serotonergic receptors. But you can see that we've looked at a variety of other targets, nicotinic receptors, retinoic acid receptors, and so on. But today I'll focus on muscarinic. So what did the Drug Discovery Center bring to the table as far as I was concerned as a medicinal chemist? On the left, I have prior to the Mulder Center Drug Discovery, or Mulder Center for Drug Discovery Research. And what I was doing 10, 15 years ago was pretty much what most academic medicinal chemists were doing, which was primarily structure activity relationship studies, right? Looking at the molecule, how it acts at our target, whatever it might be, an enzyme or a receptor, and then ultimately hoping to get into animal models. And what the Mulder Center kind of brought along was the ability to do structure property relationships as we were doing the SAR. And, you know, as we all know how important the physical chemical properties of drugs are, right? They determine all sorts of different things. You know, here are listed. I'm not going to go through all of them, but solubility, permeability, they can affect metabolism, protein binding, and so forth. So, you know, there's many properties to consider. And until the Mulder Center came along, we were really not able to readily look at those properties very effectively. So it was a big deal for us. I mean, everybody's heard about Lipinski's Rule of 5. You're probably tired of hearing about Lipinski's Rule of 5. But you know, we're all aware of it, but tracing those physical chemical properties as we do our SAR, at least in my environment at Temple University, was challenging prior to the Mulder Center coming into being. So again, just to emphasize this fact, what we were doing was looking primarily at the biological activity of our compounds, looking at the binding or the enzyme activity or so forth. What was lacking in past years was looking at the permeability, lipophilicity, polar surface area, and so forth. And also PK. We're able to do PK on a routine basis now, which was something that wasn't the case. So, you know, we're not alone. Mulder Center for Drug Discovery Research is one of many academic drug discovery centers in the U.S. I list some of them here. And that was born out of the fact that things have changed, as I'm sure you're aware, in the pharmaceutical industry and what their expectations are and what they're doing in-house. So there's a lot of opportunity in academia, and Temple is trying to capture some of that, those opportunities by having this Mulder Center available to us. This is just some of the capabilities we have. Of course, in my case, we're primarily interested in medicinal chemistry, but there's a whole host of other things that are going on in the center, in the department, that support our medicinal chemistry efforts that just weren't around or weren't available to us readily without going outside and collaborating and so forth. Now we're able to do a lot of this stuff in-house on a regular basis. So I don't need to go through all of these things. I mean, I think you all know what happens in a medicinal chemistry program, but what is different, at least in my case, was that we're able to actually do high throughput screening. We have the platforms to do that. We have libraries that we can screen, which identifies hits, and then we can take those hits, turn them into leads, and then of course go into lead modification. So again, it's just really improved our ability to attract funding. It's improved our ability to attract collaborators. And it's just been a big plus for me as a medicinal chemist to have this availability. So this is just going back to that slide that I mentioned. We're now doing SAR and SPR routinely, simultaneously, as we proceed with our projects. And as I said, this was not possible. For instance, when Richie was in the lab, we would talk about and think about the properties of the drugs that we were making, but we really didn't have a good way of looking at those properties. All right, so that brings me to the focus of my talk today, which are these lactones that we've been working on. I've been working on these simple alkyl substituted lactones as anticonvulsants since my days as a postdoc out in St. Louis at Washington University. And this project has kind of evolved over the many years. I went to Temple sometime later, worked on those lactones as anticonvulsants, followed up on it. They turned out not to be very good. The ones that we looked at later, they weren't very good as anticonvulsants, but they turned out they had some muscarinic properties. So they bound to muscarinic receptors, and that led us down a path which involved Dr. Bandare and some other graduate students. They worked on that for their PhD projects to synthesize and evaluate a variety of these arylpiperazine derivatives. Right? So that ultimately led to the final project that I'll talk about today, which was serotonin and ligands for serotonin receptors. And again, I want to tie in the Mulder Center and how that helped to contribute to our ability to expand what we were doing. So I'm going to start off looking at those muscarinic leads, the muscarinic ligands, and then go into the specific approach that we took. We did what most medicinal chemists would do. We took the molecules, our initial leads, broke that molecule up into pieces, into sections, and then we started modifying the various sections in a somewhat methodical way in order to see what the changes in the, not only the SAR, but the SPR at a later time. Right? So again, we'll get into that. And then I'll end today with the project that is targeting serotonin receptors. So this project began quite a number of years ago when one of my colleagues in the department who is interested in addiction came to me and alerted me to the fact that the NIH had just sent out proposals or announcements that they were looking for proposals, for grant proposals around addiction and whether or not I had any ligands in my laboratory that we could use to write a grant around that topic. And I knew I had these muscarinic ligands, and I looked into the literature, and it turned out that M5 had been associated, M5 antagonists had been shown to have some efficacy, at least preliminary data regarding addiction. So that kind of sent us down this path, looking for muscarinic subtype 5 receptor ligands, right? M5 selective ligands. At the time that we started this project, there were no crystal structures for the receptors, for the muscarinic receptor subtypes. That changed right around the time we were finishing up. Richie and Rong, two of my graduate students, were finishing up. The crystal structures became available, but at the start of the project, they were not. So we had to do what many of us often do, and that is just take our leads and start to modify them. And this is kind of how we went about that. This all started, as I said, when we realized that some of our lactones had might have some anticonvulsant, or some antimuscarinic activity. That was based on a report in JMed Chem by Kaiser et al., where they reported these compounds that I show at the top, these Kaiser lactones. We had these canilactones, mylactones, and we thought, well, these are all very similar compounds. Let's make some derivatives and see if they turn out to have antimuscarinic activity. And it turned out they did, and you can see at the bottom, when we made these N-arylpiperazines derivatives, they turned out to have activity. So we went ahead, we made a series of those compounds, and this is some of the initial data that we generated. And these are percent inhibition for muscarinic receptors here, right? So these are just percent inhibition at 10 micromolar. And what we found was that some of these compounds actually did have activity at muscarinic receptors. This is 82% inhibition. So we took that as a positive sign. We went ahead and thought that we'll look at these molecules, and maybe through some lead modification we can come up with some M5 selective ligands. So that was our ultimate goal. And what I want to talk about is how we went about doing that. Right? So as I said at the beginning, we took that backbone, right, the N-arylpiperazine substituted lactone. We divided it into 4 different regions. Region 1 the arylpiperazine region, primarily the aromatic ring. Region 2 is the substitution at the alpha position next to that carbonyl. We changed the groups at the position alpha to that carbonyl, as well as we moved them over to the beta position and tested those as well. Region 3 was the linker region. We changed the length of that from N=1 to N=3. And then region 4 was we were interested to see whether that carbonyl of the lactone ring was required or whether we could do with the furan derivatives. So this was basically our thought process going forward. I'm not going to talk about all of them, but I did want to show you that we did come up with chemistry. Actually, Rong and Richie came up with chemistry that allowed us to modify each of these different regions. Here we have in the upper right-hand quadrant, we have the chemistry that was used to make the aromatic substituted compounds, as well as a more efficient route to the lactone. I'll talk about that today. I'll also talk about some of the changes to region 2 that we made. I won't have time to talk about the beta substituted compounds, but I'll talk about some of the changes we made to the diethyl substituents that are out there. And again, we had chemistry that allowed us to make homologs, N=1 at the bottom, N=3. And what I'll be focused on today primarily are the N=2, because they turned out to be among the most potent of the compounds. And of course, we had chemistry to get rid of that carbonyl that I mentioned to give us those furan derivatives as well. So I'll be using this chemistry. I'll be talking about some of it. Some of it I won't have time to talk about. I just wanted to show that we had worked out chemistry to make modifications to all of those areas. All right, so our hypothesis is that it's possible to improve ligand affinity, and then later we were hoping to make inroads as far as selectivity through systematic modification of these different areas of the molecule. And as I said, we started with region 1, the aromatic ring. So, as all medicinal chemists will do, we broke the molecules apart and figured out the best way or one of the most efficient ways of synthesizing them. So we thought that if we could come up with this lactone intermediate, lots of arylpiperazines are available. Commercially, you can buy them, and then we'll see later that we ultimately started synthesizing some of these arylpiperazines. So we went ahead and came up with this process that worked pretty well for us to synthesize our target arylpiperazines at reasonable yields. Not great, but good. Good enough for us to make a whole series of them. So we went ahead and did that, and I won't go through all of this data, but the take-home message here is basically what we learned was that it seemed to us, based on the preliminary data, that ortho-substituted aromatic rings were favored here, right? Again, we were looking at percent inhibition. These higher numbers suggested to us that the ortho position was favored, so we thought that it might be a good idea to go and make some ortho-substituted arylpiperazines and test them and see how that worked out. So at this point, it was Rong who went off and thought, okay, well, let's make some substituted arylpiperazines, and one of the compounds that we found to be very good in our assays, preliminary assays, was this isopropyl derivative, right? Percent inhibition of 96%. At the time, that was one of the best compounds we had based on our preliminary data. And I should also mention that we were obtaining this preliminary data at CEREP, which is a CRO that we had to pay for this data. So it was actually fairly costly to get these compounds. Just this preliminary data, this percent inhibition data, was fairly costly. So we had that restriction as we move forward with the project. But Rong set out to make this compound in big, in larger amounts, and he ran into problems using the current, or one of the most commonly used approaches to the synthesis. And you can see that The isopropyl, you can make the compound at about 12% yield. When he tried to make the T-butyl and the iodo derivatives, he wasn't able to get any of the the desired compound. So Rong set out to improve this process, and he did a really excellent job in doing so. What he did was he evaluated the reaction that he was working on that was giving us the poor yields, and worked on ways of solving some of the problems. So in this current process that he had been using, he thought, well, we have poor leaving groups, right? These chlorides, they're leaving groups, but they're not the best leaving groups. You have an unprotected amine that can cause mischief that you probably want to avoid. You have a high boiling solvent, which again can cause issues. And on top of that, you have a long reaction time. So there's lots of factors that are leading to these poor yields in that reaction. Rong's solution was to use a nozzle protecting group on both the alcohols and the amine. So that covered all the protecting groups. He used microwave assisted chemistry to increase or decrease the reaction time, and he used acetonitrile as base, as solvent. And the reaction went extremely well. It was very productive. You can see here that Rong was able to make a variety of ortho-substituted compounds. And again, remember, we thought that by doing this, we were going to get compounds with higher affinity. But you can see from this that the yields using Rong's technique that we published in that article there were quite high. Rong went out in the literature to see what others had reported, and this is just showing you a slide that Rong was rather proud of, that showed that his method was superior in yield for specific compounds to a variety of other techniques. Not that this was necessarily the best of all approaches, but it certainly compared favorably and gave us a very effective, efficient route to the compounds we were trying to make. So the chemistry was great. We were very happy with this. We published it, as I mentioned, and then we used those compounds to see what the ability to make them brought us. And unfortunately, as research often goes, the compound that we started with, the isopropyl compound, turned out to be still the best of all those that we tested. So the T-butyl turned out not to be as good as the isopropyl. The diisopropyl, again, not as good. So it's great that we were able to make these compounds. It answered the question we were interested in, but we learned a lot. But we were somewhat disappointed in the results. We were hoping that these bulkier aromatic systems would have higher affinity. So what we learned there was, as I mentioned, orthosubstitution is favored over para. Certain groups, isopropyl, phenyl, iodo, are preferred for the orthosubstitution. And then some compounds that I didn't show, I'll refer to them later, but we had some biphenyl substituted analogs, which showed highest affinity among the compounds that we tested. The results of that I didn't show you here, but as I said, I'll refer to that later. So now we're going to move away from the aromatic ring. We've done a lot more down there, but just for the sake of time, I want to move on and talk a little bit about the region 2 and some of the things that we did there. We modified that the diethyl substituents there, and added some different groups. So once again, we back to this isopropyl derivative. What we thought would be useful or might be interesting would be to come up with some spiro derivatives, right? Where oftentimes when you're trying to design subtype selective agents, more rigid compounds, spiro compounds, for example, can often result in more selectivity. So we thought that this would be interesting, and we needed a good way to make these compounds. So once again, Rong went off to figure out a way to do this that was more efficient than the way we were currently doing it. So this is the problematic reaction that we were using with BBr3 to convert one of the products to the desired. This is the desired product. We were using BF3 in dichloromethane to generate these two intermediates. And you can see the yield is low. It was difficult to separate the compounds, the products of the reaction. So once again, Rong set off to come up with a more efficient way. So looking at the mechanism, you have the use of BF3, and you have this cation that's being generated, and it's really nothing available to quench it very effectively. So he went off to look at more efficient ways of dealing with this cation intermediate and also the unprotected hydroxyl group that was present in this reaction. And the results of that are shown here. So he came up with a modified Prinz reaction. Of course, what I just showed you is also a Prinz, but this technique worked out much better for us. Here he was using sulfuric acid as catalyst instead of BF3, and he used acetic acid as solvent instead of dichloromethane, and solved both of those problems by doing so. So the cation is being captured by the acetic acid to generate this intermediate. You have the alcohol is protected, and it can go to either one of these intermediates, and you might wind up with both of them. But we were able to actually isolate this caprolactam here, this ring system. So we believe that this is the way that the majority of the reaction goes, and then that is treated with sodium hydroxide to open it up, and then upon treatment with acid, it closes to give us our desired product. So again, the reference for that chemistry is shown there. And this is something we use still to this day. We use this to make lactones. And you can see once again the yield much improved over the previous method. The reaction is far superior than what we were accustomed to. So we were able to make these compounds in fairly good yields. And this is just a percent inhibition of those different compounds. And you can see that as we go from the diethyl, which is what we started with this diethyl substituted lactone with the isopropyl at the ortho position. That and these other spiro derivatives all kind of max out. The only one that doesn't bind very well at all is the diphenyl derivative, compound 7, which just didn't bind very well. So once again, we were able to synthesize these compounds, test them, and then we went ahead and looked at them as far as their selectivity was concerned, right? Because remember, we're trying to design or develop M5 selective agents. And this is compound 19, 21, 28, 33, and 31. Here's some of those biphenyl compounds that I referred to just a minute ago. 3 of them are shown here because they were among the higher affinity compounds. There's our isopropyl derivative, both as the diethyl and as the spirocyclopentyl. And what we learned is that our compounds were not, at least these original series, were not selective for M5. If anything, they seem to prefer, and this has been supported with later data, they seem to have some preference for the M2 receptor, right? This brown colored bar, and M4, which is the this purple bar. So again, disappointing. Even though we were able to make these compounds, we wound up with data that we were rather disappointed in. But again, we still have lots of work that we can do with these compounds. So it's not that we gave up on the compounds, but we began to look at other things. We're in the process of writing papers. We still haven't published some of that stuff from those several years back. And as I said, we did make other changes. We've changed the linker region from 1 to 3. We've gotten rid of that carbonyl. We've moved these alkyl groups from the alpha position to the beta position to test what the effect of that would be. And I showed you the chemistry that we developed to do that. So there's a lot of work done on the compounds, and a lot of compounds that were generated as a result of both Richie's work and Rong's work. And we're still following up on some of those compounds to this day. But at this point, in the interest of time, I want to switch from this project that I've been talking about, the muscarinic project, and move into the muscarinic side of things. And this is about the time, it was about 2010, 2011, that the Mulder Center came along, and the individuals that were part of the center, we started this collaboration with the psychoactive drug screening program, which is shown here on the slide, availability of PDSP. PDSP is the psychoactive drug screening program. It's supported by the NIH, and it generates lots of data for academic institutions as well as small companies all over the country. So we started getting data through the PDSP as a result of some collaborations that were ongoing at the Mulder Center. So we started getting this data, and here's some of it. So before, I told you we were getting this, paying for data from CEREP, and it was actually quite costly. So that data that I just showed about the subtype selectivity was actually quite expensive to obtain that data. This data was being supplied free of charge by the psychoactive drug screening program. And you can see, instead of just getting percent inhibitions, now we're getting, these are KI values, right? Which are, of course, much more useful. And here again, you can see that our compounds are seem to have a preference for M2 and M4, less so for M5, right? So again, not great news. But again, we haven't given up on these compounds as they relate to the muscarinic receptor. But in the interest of time, I want to move on. So the Mulder Center comes along, and now we're able to do what I started out talking about, SAR and SPR simultaneously. Here's some of the data that was supplied by the Mulder Center for Drug Discovery Research at the top, right? You can see we're getting C-log p-values, TPSA you can calculate, so that's readily available anyway. But we're now getting solubility data, rat liver microsome, mouse liver microsome, human liver microsome data. We're getting CYP enzyme inhibition, whether or not these compounds are going to affect the CYPs, and then, of course, if they affect CYPs, then you have the potential for drug interactions and so forth. So now we're getting this data routinely, and we can look at it, compare it to our SAR, and of course make more informed decisions on how to move forward with which compounds, right? So that brings us to the fact that the psychoactive drug screening program also has the ability to screen for a wide range of targets. Serotonin receptors, dopamine receptors, muscarinic receptors, GABA receptors, on and on and on. So we asked them to look at some of our compounds, some of these lactones. We had many of them. And they did. And what you can see at the bottom is these are all serotonin receptors, and the affinity, these are IC50 values. And we can see that many of them don't bind to any of these serotonin receptors, but some of them, several of them, bound very well to one particular subtype, right? And you can see that there's some that have multiple, that hit multiple receptors, but there's also some that have fairly good selectivity for the 5-HT7 receptor. So we looked at this and we thought, well, is this a new research direction for us? We're really not having very good luck with the muscarinic receptors as far as these compounds are concerned, based on what we had tried. Of course, we could try other things, but these compounds look like they're pretty selective for 5-HT7. Maybe it would behoove us to pursue this. Question was then, well, what disorder do you look for, right? What evidence do we have that these compounds are going to be good for any type of disease state, right? And if there is some indication, is there compelling evidence, and then is there an animal model that we could use to look at? And what we decided to start looking at was inflammatory bowel disease and 5-HT7 receptors. And so the next couple of slides are designed to try to convince you that 5-HT7 might have something to do with inflammation, right? Which is not something we typically associate with serotonin, right? So Crohn's disease and ulcerative colitis are major categories of inflammatory bowel disease. It's a very common problem throughout the world, and the treatments are shown here, right? There's some immunomodulatory agents, there are some anti-inflammatory agents, steroids, antibody treatments, but there's really downsides to all of the current treatments for the disease, and there's really no way to cure the disease currently. So there's definitely a need for improved treatments for Crohn's disease, ulcerative colitis, and we thought that maybe 5-HT7 might be useful because there was evidence in the literature that it had a role to play in inflammation and might affect inflammatory bowel disease. So we went ahead and started looking at this further, right? So as I said, serotonin, turns out, plays a critically important role in the gut. Right? I mean, we know that there's serotonin in the brain. We're familiar with many of the drugs, serotonin selective reuptake inhibitors used for depression and so forth. But actually, most of the serotonin that we have in our body is in our gut. 95% of it is, right? And there's cells, cells in the GI tract and enterochromaffin cells, which produce serotonin with the enzyme tryptophan hydroxylase 1, right? So people have done studies, and I'll talk about some of them in a minute, where they've shown that serotonin signaling is actually altered in inflammatory bowel disease. And they've shown that there's increased numbers of enterochromaffin cells and therefore an increased concentration of serotonin in the GI tract of patients that have Crohn's disease and ulcerative colitis. So the 5-HT7 receptor is also present in the GI tract, and it is a GPCR that's of course activated by serotonin. But the interesting thing is that it's highly expressed in dendritic cells in the GI tract, right? And due to that, some people have looked at the possible role of 5-HT7 in the immune response in the GI tract, and come up with some pretty interesting results, and I'll talk more about that in a minute. So GI enterochromaffin cells and dendritic cells, as we see in this cartoon, are co-located, right? Here we have enterochromaffin cells. We see that serotonin is being released. We have the dendritic cells with serotonin 5-HT7 receptors expressed on the surface. So the serotonin, high concentrations, remember, right? Higher than normal concentrations of EC cells, higher than normal concentrations of serotonin, stimulating 5-HT7 receptors, resulting in an increase in pro-inflammatory cytokines and an increase in the immune response, right? So again, if this evidence is compelling, it would suggest that 5-HT7 antagonists might be useful in the treatment of this inflammation of the GI tract. And that's what began our project as far as the use of our compounds as potential treatments for IBD. So the disease or disorder that's been linked to the new target was IBD. Evidence does exist, that correlates changes in the target to the disease state. There are animal models that are available, and what I'm going to show you in just a minute and I hope I still have a little bit more time, that there is, I think, compelling evidence that implies 5-HT7 role in the disease. I'm going to show you some knockout mice where they knock out the enzyme that synthesizes 5-HT7. They've done knockouts where they've knocked out the 5-HT7 receptor, and they've used 5-HT7 antagonists to show that the 5-HT7 receptor might be involved. So the model, this is just kind of a summary of what I've discussed so far: 5-HT7 synthesis, 5-HT7 receptor, and the IBD models, right? So those are three things that I'm going to present here in just a minute. Something that you may or may not be familiar with is a DSS model of IBD. This is a fairly simple model. It's an irritant, sodium sulfate, which is a chemically induced model of IBD. The DSS is just simply added to their drinking water, of course, in a specific concentration. And DSS is toxic to the epithelial cells and disrupts the barrier and leads to the inflammation, which is mimicking the effects of that we see in IBD. And of course, there are other models, more advanced models. We've done some of that too, but I want to start with this, the simplest one that's actually very commonly used as well. All right, so here's some of the evidence. At the top, we have the THP knockout mice, and remember that's the enzyme that synthesizes serotonin. And we're looking at cytokines. So this is interleukin 6, interleukin 1 beta, and TNF alpha. And what we see here is with DSS treatment, the animals that are lacking the enzyme have significantly less interleukin 6, significantly less interleukin 1 beta, as well as TNF alpha, right? So you can see really knocks down the cytokines in those assays, right? So that's the enzyme knockout. Then we have receptor knockouts, and we see similar trends here, right? With interleukin 1 beta being knocked down with the DSS treatment, the TNF alpha being knocked down with DSS treatment, with the knockouts after DSS treatment. And then interleukin 1 beta. Again, here we have the DSS animals, and we have the knockdown of the interleukin 6 levels in those animals. So again, at least consistent with the fact that serotonin and the serotonin 5-HT7 receptor may be playing a role in the inflammation in these models. And then finally, they did a 5-HT7 antagonist. This was one that was available for many years by, I think it was SmithKline Beecham reported this 5-HT7 antagonist some years ago. It's a fairly selective antagonist, and they used it in the same DSS model. And I think you'll see, we would agree that we get similar results as the knockout animals, where we have the interleukin 1 beta, TNF alpha, and interleukin 6 being significantly reduced in the DSS animals after the administration of 40 milligrams per kilogram of that particular 5-HT7. So again, I think this is, here's some look at the histology showing with vehicle. You can see that the tissue is pretty well inflamed and not looking healthy. Here with the treatment group looking somewhat better after a 5-HT7 antagonist. So we saw that, at least in our eyes, we felt that this evidence supported further investigation and that 5-HT7 antagonists may indeed be able to slow the progression of this disease. So again, we found this, thought this to be compelling evidence regarding the role of serotonin and 5-HT7 in IBD. This is just a summary of the different things that I just presented: knockout mice of the enzyme, the receptor, the use of the 5-HT7 antagonist. So now the question is, will our compounds be able to do what we just saw the SmithKline-Beecham compound do? So we started out looking at the data. This is some of the data that I showed earlier, showing the selectivity. And if we look at these various compounds, we chose what I call here is compound 10 as our lead compound, simply because it was most, well, not just because it was most selective, but that was one of the important reasons. It also had some some useful PK properties and ADME properties. If we look here, compound 10 had a half-life in mouse liver microsomes of about 18 minutes, which suggested that it would have a reasonable half-life in mice. And then it was not active in the CYP. It did not inhibit CYP enzymes, which again is a positive finding. So we went ahead and used that as our lead compound. And this is just summarizing some of the properties of it. It's a potent antagonist. It's a functional antagonist, 18 nanomolar functional activity. It's highly selective compared to other 5-HT7 receptors. And as I just mentioned, it's stable in mouse liver microsomes. It's soluble. So I'm sorry that it keeps progressing without me telling it to. It's soluble and doesn't have the activity. So it had all the properties, or at least had many of the desirable properties that we were looking for. So we went ahead and looked at the PK first. And here we can see IV dosing in mice, 5 milligrams per kilogram PO in mice, and half-life was about 3 hours. Bioavailability, about 17.5%. So we were actually pretty happy with all that. And we felt that we were ready to go on and do some in vivo work in mice using this acute BSS mouse study. So I'm going to show you some acute BSS studies, as well as a chronic DSS study. And here, the BSS is administered in the acute model for in days 2 to 6. In the chronic model, you'll see it's longer. What we found in our studies were not unlike what I showed you earlier, not maybe quite as pronounced, but we did see positive effects and significant effects on TNF alpha, interleukin 1 beta. You can see that the compound we're calling 073 here knocks down the interleukin 1 beta. And then if you look at the histologic scoring, macroscopic scoring, again, significant reductions in those scores. So we felt that our compound produced an improvement in inflammatory cytokines, the histologic score, macroscopic score, and then the disease activity index, which is kind of a compilation of many different factors, was reduced as well. And again, at the bottom, looking at the histology, we see that with saline, we can see a clear deterioration of the tissue, whereas with treatment of our compound, the tissue looks reasonably healthy when compared to control. So this provided some proof of concept for us, the acute model, and then we went on to look at the chronic model, the chronic DSS model, which, as I said, is done over an extended period of time and a longer period of time. And we got, just showing you histologic scoring and macroscopic scoring here, we did get significant reductions in those scores, again, suggesting that the compound was having a beneficial effect in the chronic DSS mouse models. So I think I'm still okay with time, but I am done with my talk. I just want to summarize some things and acknowledge some contributions of different people. The summary, our aryl piperzinyl compounds are competitive 5-HT7 antagonists with drug-like properties. We've developed very efficient methods to synthesize these compounds, and we've come up with a wide range of structurally diverse analogs. Our work suggests that the series has potential as IBD therapeutics, and we have proof of concept of the lead that I just talked about in acute and chronic DSS models, and further work is underway in other models of IBD to further investigate the potential use of these compounds for that particular indication. So these are some of the I mentioned at the beginning that we have the Mulder Center for Drug Discovery Research there at the back. And I'm currently the interim director of the Mulder Center for Drug Discovery Research. I've been in that position for only a couple of months now. But I've been involved with the center since its inception. And these are some of the individuals who contributed directly to this project. Heidi was a master's student in the lab. Doug Pippin is the CEO of a startup company that resulted from this work. Doug Pippin is the CEO of Preventix, that I mentioned here on the slide. It's a startup company based on intellectual property surrounding these compounds as leads. Kevin Blattner, right next to Doug, he was actually a graduate student during the beginning of this work, and Kevin now works for Preventix. He's a scientist working for the startup company, working with Doug to further this project along and see what will come of these compounds. Ben Blass is a collaborator who's been involved in the project, the serotonin aspect of the project since the beginning. So as I mentioned at the bottom here, Preventix is focused on novel cytokine modulators. That startup was in 2015. We've had a history of NIH funding for the past 5 years. We currently have a phase 2 SBIR, and we're submitting, September 5th, we're submitting 2 more grants. And I mentioned a PhD student, Kevin, was hired recently by Preventix. And these are the acknowledgments of my colleagues in the Department of Pharmaceutical Sciences. Ben Blass, an important contributor. Brian Day is a new graduate student. I mentioned Heidi. Ron Gao and Richie Bandari made important contributions way back 2013 and 2012. It doesn't seem that long ago, but they both contributed significantly to these projects, and then the other people were present around for much of it. Ken Krzyzekwa and Min Yi made valuable contributions to the PK studies that I presented today. The Dean's Office was involved, Peter Dukas, in supporting some of this work, financial support. The Mulder Center, Jack Gordon is the guy who does the screening facility work, the ADME work, provided a lot of the data that I showed you regarding the SIPs and the mouse liver microsomes and so forth. Marlene and Dennis were contributors to the pharmacology. Wally Khan was the guy who first reported that 5-HT7 might be useful in IBD, and we followed up on that work. We actually collaborated with Dr. Khan. We just finished a grant, a 3-year grant with the Crohn's and Colitis Foundation, and then some, which I mentioned down here, and then some of the other support that we've received over the years. One of the drawbacks that I did want to mention, being from an academic environment, I spent most of my career trying to generate data so that I could publish it. Being part of a startup company changes that somewhat. So you have to always be concerned with intellectual property, confidentiality surrounding that intellectual property. So there's costs involved in preparing the patent applications that have to be considered. And then you have to make sure that you're you're filing those patents in a timely manner so that eventually you can publish your data. But it definitely slows the rate of publication. I have not published in this area for a number of years, and it's not because we don't have data. I showed you some of the data, but we have a lot of data that we could publish that we're just sitting on for the moment due to intellectual property concerns. So there are some drawbacks, but there's also a lot of positive things that have come out of it. These are just some of the papers and things that we've published. Some of the patents that have come out of this work are shown here as well. But for now, I'd like to thank you all for giving me this opportunity. I really enjoyed it. Was glad to support your efforts, to support Dr. Bhandari. Always good to see him. So with that, I'll open things up for questions, and I assume I'll stop sharing at this point. Thank you.
Thank you, Dr. Kenny, for your outstanding talk on lead modification approaches for GPCR ligands. So I'll open the forum for some questions. Audience, if you want to ask any questions. Any questions? Yes, I can't see any question from the audience. So in that case, I have few questions for Dr. Daniel. So
Okay.
the first question is, why do you choose those aryl piperazine lactones as a GPCR ligand?
Well, the original reason for choosing them, as far as the muscarinic pathway was concerned, had to do with that paper that I mentioned, the Kaiser lactones, right? So that was many years ago, but that's what really first alerted us to the fact that those compounds might have activity at muscarinic receptors. Why we went with arylpiperazine specifically as a the amine containing portion, right? Because prior to that we had other amines on there. But the reason we chose the arylpiperazines was the fact that there were, if you look at serotonergic ligands in general, if we were going to turn from muscarinic to, or even in the muscarinic field, arylpiperazines are very commonly found in muscarinic ligands. They're found in serotonergic ligands. It's just a very commonly used and it's a commonly used fragment, and it also allows you to do a lot of things on the aromatic ring that can lead to more information as far as structure activity relationship is concerned. We were, in the past, we had as our amine just a heterocycle or diethylamine or something like that. So what we were interested to do was to keep our alkylamine, but also add an aromatic substituent, right? Which we hadn't done in the past. So that was one of the rationales that we used.
Okay. So there is another thing that I observed, that you said the orthosubstitutes are preferred over the parasubstituted ligands. So
Right.
what may be the reason? Have you seen the docking studies? I think for GPCR it is a little bit difficult to do the docking studies.
We have not done any, well, we're in the process of doing some, but I don't know what the results of those are going to be. But we don't really understand exactly what's going on with the ortho substitution, but we believe that it has something to do with the relationship between the aromatic ring and the piperazine ring, and how it's oriented is favored by the receptor, and depending on what you have at the ortho position, it's going to change that orientation, right? Something at the other end at the para position is not going to really affect that relationship, right? The piperazine and the aromatic ring are going to be able to do whatever they want as far as the para position, but ortho is going to sterically affect that. Excuse me.
Yes, that can be a possible reason. Yes. So, yes, any other question from the audience? Someone has asked why from 5-HT serotonin receptor, which are autoreceptors. So I do not understand exactly what you want to ask.
What? Say that again?
He's asking from 5-HT serotonin receptor, which are autoreceptors.
I still didn't quite hear I heard autoreceptors, but I didn't hear the rest of the question.
Yeah, she's asking from 5-HT serotonin receptor, which are also autoreceptors. Autoreceptors. You can see the chat box if you want, but I do not understand.
Yeah, I mean, there's a lot of the compounds that we're dealing with here are going to hit primarily 5-HT7, and let me see if I can see what the chat is. Maybe. Oh, yeah. The question has to do with which of there are a number of different, I forget how many serotonin subtypes there are currently, but the question has to do with which ones are autoreceptors. And I have to say that I don't know with confidence which ones are autoreceptors, but in this case, in our compounds, have these receptors that we're interested in and focused on are those that are on cells of the immune system, right? Like dendritic cells. 5-HT7 is also present in other cells of the immune system. But as far as the effect on autoreceptors is concerned, I really can't comment on that. I'm not sure.
There is one more question. Have you studied either in silico or in vivo metabolism of the best compounds, as in compounds with promising activity?
We have not done that. That's something that we've discussed, and it's something that we will probably have to do, or we certainly should do it, but it's not something we've had the time to do at the moment. Right now we're having problems figuring out how to get enough of our compounds into the tissue of the GI tract. So we have discussed the need for a better understanding of the metabolism of our compounds, but we've not done any of that in silico work at this point. But it certainly would be very useful information to have.
Yes. So I must thank you, Dr. Kenny, for your outstanding informative talk on lead modification approaches for GPCR ligands. So I think it is early morning in Philadelphia, and you have quite managed well your time to deliver such a nice talk to our audience. We are truly honored. Thank you very much for joining us. And thank you very much.
It was a pleasure.
Thank you so much. So
thank you.
Okay.
Thank you for the invitation.
Yeah, I will always have you in our scientific deliveries. So thank you very much for joining us. And with Dr. Kenny's talk, we'll conclude today's webinar. We'll join tomorrow. Thank you very much.
All right. Thanks. Take care.
So tomorrow